YouTube channel. So today we are going to talk about meiosis. Meiosis is a cell division process whereby one cell divides to form four daughter cells. And in this case, the four daughter cells are haploid. They have the number of the chromosomes as the parent cell. So if the parent cell has 46 chromosomes, the daughter cells will have 23 chromosomes. So they are haploid. So I take the word ha to have instead of ploid so that I can understand it better. So the question is, where does meiosis take place? So meiosis take place in gametes. And when we say gametes, we are referring to the sex cells. Unlike mitosis, where it takes place in somatic cells. Somatic cells are the body cells, but gametes are the sex cells. So the sex cells in females is ovum, or rather the egg cells, and in males is sperm cells. Yeah. And we know that we cannot study something for the sake of studying it, or we cannot just do something for the sake of doing it. We need to ask ourselves the importance of whatever that we are talking about, or rather, where is it taking place when we talk about biology. So the importance of meiosis is that it is responsible for the formation of the sex cells. The sex cells are what we call gametes, and it also activates the genetic information of the sex cells. And this happens because of the crossing over, whereby the chromosomes are crossing over to exchange genetic material, and it maintains the constant number of chromosomes by halving the cell. So the crossing over produces a combination of traits and variations. So during crossing over, it's where genetic material is exchanged. So now I speak about the ovum and the egg cells in females, which are the gametes, and then in males it's only the sex cells. So yeah, it's only one word for the males, so it shouldn't be confusing. So now we have to talk about the phases of meiosis. The first phase is interphase, the second phase is prophase, the third one is metaphase, the fourth one is anaphase, the fifth one is telophase and cytokinesis. So the best way to remember this is to say IPMAT or IPMAT. Then you know that IPMAT stands for something. Some books argue that interphase is not part of meiosis, but I'll give you the reason why we include the interphase stage. So interphase is the site for DNA replication, meaning that DNA replication takes place in interphase. And we also spoke about the exchange of genetic material. So DNA replication, we know that it's a process whereby a DNA makes the exact copy of itself. So this stage is important for each cell before the division. So it's important to double check the genetic material before the division. So interphase it also grows chromosomes. It also prepares the cells for the cell division. That is why I'm saying this is going to be our first step for the meiosis phases. So during prophase, you can see visible chromosomes. You can also see a centriole. You can also see spindle forming, and then you can also see the chromatids. So obviously when you see chromosomes, you can see the chromatids because chromosomes are made of two chromatids. So the starting cell is diploid, and the chromatid condenses to form chromosomes. As you can see, visible chromosomes, and homologous pair come together in pairs. So our chromosomes are going to pair. So we also have crossing over. So when we talk about crossing over takes place during prophase. Please remember that crossing over takes place in prophase. DNA replication takes place in interphase. Always remember that. And then we also have the nuclear envelope breaking down. So the nuclear envelope we spoke about. So the nuclear envelope is represented by the broken lines, or the lines that is surrounding the chromosomes. So that is our nuclear envelope. During metaphase, homologous pairs align along the center of the cell. So the center of the cell, we call it equator. Remember this, homologous pairs, or you can say chromosomes pairs align along the equator. If they ask you what happens during metaphase, and then we can also see the spindle fibers attached to the centromere. So the spindle fibers attached to the centromere, and they also attach to the central. And we also have a metaphase plate. Metaphase plate is our equator. So during metaphase, chromosomes align along the equator in homologous pairs. That is the most important thing that you need to remember when it comes to metaphase. So during anaphase, the homologous pairs which were aligned along the equator, they are separating, so they move to opposite poles. And then we know that chromosomes is made of a sister chromatids. Sister chromatids are the chromatids that are combined to form a chromosomes. So they are attached to the centromere. Now we're speaking of chromosomes. These chromosomes separate. Remember, they were together along the equator. Now they are moving separate ways. And our last step is telophase 1. So in telophase 1, the chromosomes gather at the poles of the cells. Remember, they separated, or they were separating during anaphase. Now they are gathered at the poles, or let's make it simple, they are gathered at opposite poles, and the cell divides via cytokinesis. So when you see the cleavage, where it's written cytoplasm starts to divide, it means cytokinesis is happening, because cytokinesis is the division of a cell, and the nuclear envelope reforms. The nuclear envelope is the broken line, that is the nuclear envelope. So we also have nucleoli over nucleolus. So the nucleoli, usually it's represented by a dot inside the nuclear envelope. So meiosis happens in two phases, or rather in two forms, meiosis 1 and meiosis 2. So in meiosis 1, we've included interphase, whereby DNA replication takes place. However, in phases of meiosis 2, we are not going to include interphase. We are going to start with what? With prophase, because we are continuing with the cell that has divided to form two cells. So now we are going to continue with our two cells. Whatever the other cell do, the other cell does. So it's like what you do with mathematics. What happens on the left hand side must happen on the right hand side. So this is what we are going to focus on when it comes to the cells. We are going to continue with two cells instead of one. So during prophase 2, the spindle fibers forms, and the chromosomes and nuclear envelope are visible. In prophase, you can see visible chromosomes. You're not going to see the chromatids. So the chromosomes and nuclear envelope, they are visible, and there is no crossing over. Crossing over took place in prophase 1. In prophase 2, there is no crossing over. If they ask you a question during an exam and they ask you what is the difference, or tabulate the differences between prophase 2 and prophase 1, this is the most important thing. There is no crossing over. And you must also include that the chromosomes are not in pairs this time around, especially in prophase 2. No crossing over, and homologous pairs, no. It's only just chromosomes that are visible. But in prophase 1, you're going to have your chromosomes pairing up, you're also going to have your crossing over taking place. So in here you can see chromosomes, you can see nuclear envelope that is represented by the broken lines, you can see the central, you can also see the spindles which are formed. The second step of meiosis 2 is metaphase. So during metaphase, we know the most important thing, or rather the only thing to focus on, is the equator and the chromosomes. The chromosomes are aligned along the equator individually. Unlike in metaphase 1, the chromosomes aligned along the equator in pairs, in homologous pairs to be specific. So here the chromosomes are going to align along the equator individually, and we have agreed the metaphase plate is our equator. We have the spindle fibers which are attaching to the what? To the centromeres. We also have the centroles that is attaching the spindle fiber to the chromosomes. We have our cytoplasm, we have our chromosomes. So remember the stage. Metaphase 1 has homologous pairs along the equator, and metaphase 2 has individual chromosomes along the equator. During anaphase 1, we had chromosomes being pulled apart from each other to opposite poles during anaphase 1. So now in anaphase 2, we are actually separating the actual chromosome. So we are separating them into chromatids. So chromatids move to the opposite poles during this time, and they are going to form 4 daughter cells. So our chromosomes are now divided to chromatids, and the chromatids are going to form our daughter cell, because there is only the last step, and we are only going to have 4 daughter cells. Our last step is telophase and cytokinesis. So during telophase, we are going to have 4 sets of daughter chromosomes during telophase 2. 4 sets of daughter chromosomes which are haploid. They have half the number. They started with 2n, now they end up with n. So, and a new nuclear envelope begins to form around each set. If they ask you, like the question that came during the formal test for grade 12's first term 2024, what they've asked was for the learners to draw the things that is happening to the cell, or rather let me say to draw the stage, yes, the last stage of the cell division of meiosis, of which it was telophase. So they needed to have 4 cells, and they also needed to have the chromatids instead of chromosomes. They also had the nucleolus, and then they needed to have the nuclear envelope to show that it has developed, or it has reformed again. So this is the end of our lesson. What you should grab from this lesson is that one cell divides to form 4 cells which are haploid, and these cells, they are in the sex cells, or they are the gamete cells. So when we talk about the sex cells in females, we're talking about ovum and also an egg. In males, we are talking about a sperm cell. So the most important stages that I also want you to remember is that during interphase there's DNA replication, during prophase there's crossing over, and metaphase, in metaphase 1, the chromosomes align along the equator, and in metaphase 2, the chromosomes align along the equator individually, whereas in metaphase 1, they align along the equator in homologous pairs. I hope you enjoyed this lesson. Please subscribe, share, like, and comment. Bye.